Let's have now a review of the ultrastructure of the cell. So again, when we speak of the cell ultrastructure, these are the parts or structures of the cell that can be observed using the electron microscope, right? Because many of these structures are so small that they can only be examined through the electron microscope. Okay, so first we have the cell membrane. So let's have a quick review of the cell membrane. All right, now in terms of function, the cell membrane is said to be a boundary or a barrier. The cell membrane forms a boundary between the cell and the external environment. So as a boundary, the cell membrane separates what is inside from what is outside of the cell. So it acts like a barrier that separates what is in to what is outside of the cell. And if we will compare the skull to a giant cell, then we can compare the cell membrane to the perimeter wall or the perimeter fence of the skull, because again, the perimeter wall or the perimeter fence separates the inside of the skull from the outside. Okay, also the cell membrane functions as a semi-permeable barrier. So what do you mean by a semi-permeable barrier? So when you say semi-permeable barrier, there is regulation. The cell membrane only allows certain molecules to enter and leave the cell. So there is regulation of the entrance and exit of substances. The cell membrane regulates what goes in and what goes out of the cell. So that's what we meant by a semi-permeable barrier. So again, if the skull is a giant cell, then we can compare the semi-permeability of the cell membrane to our security guards. So our security guards regulates the entry and exit of students, faculty, even visitors. Alright, so that's the function of the cell membrane as a structure that regulates the entry and exit of substances. Alright, now in terms of the structure of the membrane as seen through the electron microscope, 75% of the cell membrane or the plasma membrane are made of phospholipid molecules. Alright, then about 20% of the membrane is composed of cholesterol. Then the remaining 5% are glycolipids and proteins that are embedded in the phospholipid bilayer. Alright, so the cell membrane is made up of phospholipids. Now the phospholipid molecule has a polar head. Now the polar head is hydrophilic, meaning that it attracts water molecules, and attached to the polar head are two fatty acid tails or nonpolar tails. So the nonpolar tails are hydrophobic, meaning that they repel water molecules. So that's a phospholipid molecule. A phospholipid molecule has a head which is hydrophilic and two fatty acid tails which are hydrophobic. And under the electron microscope, the cell membrane is composed of two layers of these phospholipid molecules. That's why the cell membrane is said to be a bilayer made up of two layers of phospholipids. Okay, and the phospholipid bilayer allows lipid-soluble molecules to easily enter, and very small molecules like oxygen, carbon dioxide. Now, in the case of water, though water is a polar molecule, so polar molecules generally cannot enter the phospholipid bilayer. Water molecules can easily enter the cell membrane because of certain channels called aquaporins. So through these channels, aquaporins, water can easily enter the cell through the phospholipid bilayer. 20% of the plasma membrane are cholesterol molecules. So what's the function of cholesterol? Now, cholesterol being 20% of the plasma membrane basically aids in the flexibility of the cell membrane. So that's the function of cholesterol, that aids in the flexibility of the plasma membrane. Alright, and embedded in the plasma membrane are proteins. And these proteins, they function as alternative gateways that will allow certain molecules, especially large molecules like glucose, certain lipid molecules, to cross into and out of the cell, okay, by passing through the membrane. These proteins, they serve as channels, or alternative gateways, so that these large molecules can easily enter the cell. These are alternative gateways that will allow certain molecules to cross into and out of the cell by moving through open areas or channels of the protein channel. So that's the function of these proteins that are embedded in the plasma membrane. Okay, next, let's proceed now to the cytoplasm. Now, basically, the cytoplasm is a jelly-like material or a jelly-like substance that is located between the plasma membrane and the nucleus. So, the cytoplasm is a jelly-like material between the plasma membrane or the cell membrane and the nucleus. And suspended in the cytoplasm are organelles. Organelles are structures that perform specific functions, like an organ, right? An organ is a structure that performs a specific function, like the heart. The heart pumps blood, or the stomach has something to do with digestion. So it's the same with these organelles. Now, these organelles, these are tiny structures that are suspended in the cytoplasm that perform specific functions for the cell. And these organelles, they are classified according to the following. There are three major types of organelles. So first, we have the endomembrane organelle, energy transducing, and cytoskeleton. Alright, now when we speak of endomembrane organelles, these are organelles that are surrounded by a membrane similar to the plasma membrane. So under the endomembrane organelles are the following. So we have the endoplasmic reticulum, we have the Golgi apparatus, the lysosomes, peroxisomes, and vacuoles. Now for energy transducing organelles, these are organelles that can harness the energy either from the sun or from food molecules, such as the mitochondria. The mitochondria harnesses energy using food molecules like glucose. And we have the chloroplastids that harnesses the energy from the sun through photosynthesis. And lastly, we have the cytoskeletal framework or the cytoskeleton. So the cytoskeleton provides a framework for the cell, and they are composed of microtubules and microfilaments. Alright, so very quickly, let us go into the details of these endomembrane organelles, starting with the endoplasmic reticulum. Now, under the electron microscope, the ER or the endoplasmic reticulum is composed of a network of membranous canals called cisternae. And again, just like what we mentioned, the structure of the organelle will dictate the function. So being membranous canals, the basic function of the endoplasmic reticulum is for the distribution of substances throughout the cell. Okay, and if you will compare the endoplasmic reticulum to a school, then the ER can be compared to the hallways of the school. Or if we will compare the ER to a city like Manila, then the ER will be the network of highways. Alright, so there are two types of ER. We have the rough ER and the smooth ER. Rough endoplasmic reticulum and smooth endoplasmic reticulum. Alright, now the smooth endoplasmic reticulum looks smooth because there are no ribosomes attached to its surface. Now, it looks smooth because it lacks ribosomes. The rough ER, on the other hand, looks rough because there are ribosomes attached to its membrane. Alright? Okay, so rough ER looks rough because of these ribosomes. Smooth ER, they look smooth because they lack ribosomes. And again, ribosomes, these are structures in the cell that assembles amino acids to form a polypeptide chain or protein. Ribosomes, these are protein factories of the cell, assembling amino acids to form proteins. Okay, so this is rough ER or rough endoplasmic reticulum with ribosomes attached to its membrane. Ribosomes, they are the protein factories of the cell. So, specific functions of the rough ER, since there are ribosomes attached to its membrane, the rough ER or the rough endoplasmic reticulum is concerned with the production and storage of proteins. Again, because of the presence of the ribosomes. Okay, before they are used inside the cell or are secreted to the exterior. Also, the rough ER is a membrane factory because, again, like what we have mentioned earlier, the cell membrane has proteins that are embedded in the phospholipid bilayer. And these proteins are manufactured by the ribosomes attached to the rough ER. Alright, what you're seeing now on screen is the smooth endoplasmic reticulum. Alright, the ER looks smooth because of the lack of ribosomes. Alright, specific functions of the smooth ER. So the smooth ER is involved in lipid synthesis. Alright, enzymes of the smooth ER synthesizes lipids including oils, phospholipids, and steroids. Alright, this includes the sex hormones of vertebrates and adrenal steroids. The smooth ER also is involved in the catalysis steps in the conversion of glycogen to glucose. And also the smooth ER is involved in the detoxification of drugs and poisons, which also occurs in the liver. Alright, so that's the smooth ER. Okay, let's talk about now the Golgi apparatus or the Golgi bodies or Golgi complex. So the Golgi apparatus or the Golgi complex under the electron microscope is composed of flattened membranous sacs, much like stacks of pancakes or stacks of pita bread called cisternae. Okay, so something like this. Everybody kindly take a look at the electron micrograph of the Golgi apparatus. So the Golgi apparatus is composed of at least 7 to 8 of these flattened membranous stacks or sacs or cisternae, much like stacks of pancakes. Okay, and the function of the Golgi apparatus is said to be the center of manufacturing, warehousing, sorting, and shipping. And again, to put it simply, the Golgi apparatus is said to be the packaging center of the cell. As a packaging center, it manufactures, warehouses, meaning that it stores, it sorts out, and ships proteins for excretion. The Golgi apparatus is especially extensive in cells that are specialized for secretion. Okay? Alright. So if the school is a cell, then we can compare the Golgi apparatus to the classroom. No, because in the classroom, students are modified, their behavior is modified, their way of thinking is modified. So like the Golgi apparatus, actually the Golgi apparatus is involved in the modification of proteins for packaging. So proteins from the ribosomes are then delivered to the rough ER to the Golgi apparatus. The Golgi apparatus then modifies these proteins for secretion, okay, to be exported out of the cell. So that's the function of the Golgi. Alright, next we have the lysosome. Now the lysosome performs several functions. The lysosome contains hydrolytic enzymes that breaks down molecules. That's why I mentioned here the lysosome is a membrane-bounded sac of hydrolytic enzymes. So inside the lysosomes are hydrolytic enzymes that breaks down molecules. So lysosome is said to be the digestive organelle of the cell. So these enzymes, they break down macromolecules. Right, so they are the so-called digestive organelles of the cell. Lysosomal enzymes or the hydrolytic enzymes can hydrolyze or break down proteins, fats, polysaccharides, and nucleic acid. So they are the so-called digestive organelles of the cell. So those are the lysosomes. And again, if the school is a giant cell, then we can compare the lysosome as a digestive organelle to the canteen. Now, I don't know if you can accept this analogy because in the canteen there is digestion. Since the lysosomes are the digestive organelles of the cell, we can compare it to the canteen of the school. Okay? Alright. Also, kindly imagine this. So, imagine the membrane of the lysosome rupture. So meaning nabutas yung membrane ng lysosome. So what do you think will happen? If the membrane ruptures, then the hydrolytic enzymes inside the lysosomes would leak out. No? Lalabas yung enzymes niyan. And if the enzymes leaks out, then basically it will eat up the cell. This is called autodigestion. So while rupturing one or few lysosomes, it has little impact on the cell, but massive leakage from lysosomes can destroy the cell. And we call this autodigestion or cell suicide. So once these hydrolytic enzymes would leak out because of the rupturing of the membrane, then autodigestion can occur. Now, what is the significance of autodigestion? Autodigestion is involved in developmental processes. Like, for example, I'm sure you're familiar with the tadpole, no, yung botete. So, as the tadpole matures into a frog, di ba yung tail niya nawawala? No? The tail, the fish-like tail of the tadpole gradually disappears. Now, the disappearance of the tail has something to do with these enzymes leaking out. So, we call that autodigestion in the developmental process of certain animals. Like from tadpoles, as they mature, they lose their tail. So, that's autodigestion. Another function of the lysosomes is autophagy. Okay, these enzymes, yung mga enzymes sa lysosome can break down worn out organelles. Okay, lysosomes can fuse with a worn out organelle, say a worn out mitochondrion, and recycles the organic compounds. Okay, so this recycling of worn out organelles or destroyed organelles is called autophagy. Alright, and with that function of recycling worn out organelles, you can compare that function of the lysosome to our garbage disposal system. Yung mga janitors, yung mga garbage collectors natin. The lysosomes are the garbage disposal organelles of the cell. Now, since they perform autophagy, again, autophagy is the recycling of worn out cell parts. Now, we can compare them to our janitors and garbage collectors. Alright, so those are the functions of the lysosomes, being the digestive organelles of the cell. So the hydrolytic enzymes in the lysosome can break down macromolecules, like proteins, carbohydrates, so digestive organelle. The lysosome also is involved in digesting worn out organelles, so that's what we call autophagy. Now, the organic compounds are being recycled. So digesting organelles yun, or autophagy. And lastly, the lysosome is involved in digesting its own self, autodigestion, digesting cells. And this function of the lysosome is important in certain developmental processes, like the tadpole losing its fish-like tail as it matures into a frog. Okay, so those are the three functions of the lysosome: digestive organelle, in autophagy, and in autodigestion. Alright, peroxisomes. Peroxisomes are involved in recycling products of photorespiration in the chloroplast. And glyoxysomes, also present in the plant cell, are associated with the breakdown of lipids to be used as an energy source, especially in the seeds of plants. Yeah, found in the seeds and spores of plants during germination. Okay, and lastly, we have the vacuole for the endomembrane organelles. Now, the vacuoles, these are the storage compartments of the cell. For the plant cell, we generally call it a vacuole, but for the animal cell, we call it vesicle. A vesicle. For the plant cell, the vacuole contains water. For the animal cell, vesicles may contain other substances. Alright, and if the skull is a giant cell, then we can compare the vacuole to the storage compartment, yung mga lockers. The vacuoles are the storage compartments of the cell, just like the lockers that allows you to store your stuff. Or storage tank. The storage tank can store water. Sa school may ganyan, di ba? Storage tank. So you can compare that to the vacuole. Alright, now for certain organisms like protozoans, there are two major types of vacuoles found in protozoans. We have the food vacuole that stores food, and we have the contractile vacuole that removes excess water for protozoans, like this paramecium. Protozoans like the paramecium, the amoeba, they have a food vacuole that can store and digest food, and contractile vacuole that removes excess water. Alright, and for the plant cell, we have the central vacuole that contains water, and again, water is an essential ingredient for photosynthesis. Alright, function of the central vacuole. Yan. Sa plants ito, stockpiling proteins or inorganic ions, depositing metabolic byproducts, storing pigments, and defensive compounds. Like since sa sibuyas, tama ba? Sibuyas. Also increases surface to volume ratio for the whole cell. Okay, so I guess that's it for the endomembrane organelles. Next, let's proceed now to the energy transducing organelles. So, under the energy transducing organelles, you have the mitochondria and the chloroplast. Energy transducing organelles, these are organelles that can harness energy from food molecules like the mitochondria, or energy from the sun like the chloroplast. Now, for the mitochondria converts energy from food molecules like glucose to form the cell that can be used by the cell, and that energy is ATP. So basically, glucose is converted to ATP by the mitochondria through a process known as cellular respiration. Right? Cellular respiration is composed of a three-step biochemical pathway. We have glycolysis, Krebs cycle, and the electron transport chain that converts glucose into adenosine triphosphate or ATP. ATP is the energy currency of the cell. That's why the mitochondrion is said to be the powerhouse of the cell. We can compare that to the electrical supply of the school, like yung Meralco power lines of the school. Alright, in terms of its ultrastructure under the electron microscope, the mitochondria is a double membrane organelle. It's a double membrane structure. You have an outer membrane that is smooth, while the inner membrane is folded, as you could see in our illustration. The folding of the inner membrane are called cristae. Alright, the cristae increases the surface area for cell respiration to occur. Alright, between the outer and the inner membrane is a space known as the inner membrane space. Between the outer and the inner membrane, again, is the inner membrane space. And inside the mitochondrion is a fluid-filled space known as the mitochondrial matrix. The innermost area of the mitochondrion is the mitochondrial matrix. So if we will talk of cell respiration, the electron transport chain occurs in the cristae. Okay, and the Krebs cycle occurs in the mitochondrial matrix. Glycolysis occurs outside the mitochondrion. Glycolysis occurs in the cytoplasm, if you will recall your cellular respiration. Okay, so that's the ultrastructure of the mitochondrion. So it's a double membrane organelle. So you have an outer membrane, which is smooth, the inner membrane, which is folded, called the cristae. Between the outer and inner membrane is the inner membrane space, and the innermost area of the mitochondrion is the mitochondrial matrix. Right, here is a photomicrograph, an electron micrograph of the mitochondrion. Right, it's an elongated organelle, and you could see here the cristae and the matrix. Below that is the rough ER. As you can see here, this is the rough ER with ribosomes. And above here are lysosomes. Okay, for the chloroplast, anyway, it's a double membrane structure just like the mitochondrion. So the outer membrane is smooth like the mitochondrion, while the inner membrane takes the form of stacks of coins called thylakoids. So inside the chloroplast are flattened structures called thylakoids. Stack of thylakoids is called the grana or granum. Alright, thylakoid membrane is divided into coin-like structures called grana. So stacks of thylakoids form grana. And in between grana is a fluid-filled space called the stroma. Okay, the grana, those thylakoids that form the grana can be compared to the cristae of the mitochondria. The stroma can be compared to the mitochondrial matrix. And the chloroplast performs photosynthesis, so it harnesses the sun's energy again to produce ATP and glucose through photosynthesis. Okay, so those are the chloroplasts. And last, for the organelles, the cytoplasmic organelles, is the cytoskeleton. The cytoskeleton provides shape, maintains the shape, and support for the cell. And the cytoskeleton is composed of microtubules and microfilaments. So if the school is a giant cell, then we can compare the cytoskeleton to the beams. Yung mga columns, yung mga pillars that supports the school building. Okay, let's talk about now the microtubules. Now under the electron microscope, these microtubules are straight hollow rods, about 25 nanometers in thickness. Hollow, no, meaning parang may space sa loob. Hollow rods. Function of these microtubules, they provide shape and support and help organelles move around the cell. And they also form centrioles. These microtubules, they form centrioles. And these centrioles are involved in cell division. So these microtubules are greatly involved in cell division or mitosis. But centrioles are not found in the plants. No? Centrioles are visible only in the animal cell. What about microfilaments? Microfilaments are straight, solid rods. Again, may I repeat, straight, solid rods. Unlike the microtubules are straight, hollow rods. Microfilaments are straight, solid rods. So wala siyang space sa loob. About 7 nanometers in diameter. Okay, function of the microfilaments, they function in muscle contraction. Okay, especially yung actin myosin filaments that are involved in muscle contraction, cytoplasmic streaming, amoeboid movement, and changes in the shape of the cell. So those are the cytoplasmic organelles, the endomembrane organelles, the energy transducing, and the cytoskeleton. Okay, let's talk about now the nucleus. Now, the nucleus is said to be the control center of the cell. The nucleus is the control center of the cell, and it also contains our genetic blueprint, DNA. So the nucleus is composed of the following structures. So we have the nuclear membrane, nucleolus, okay, chromosomes. And if we will compare the nucleus to a school, then we can compare the nucleus to, let's say, the dean. Right? Dean or the president of the school, or in senior high school, it's the principal. For the collegiate level, either the dean of the college or the president of the university. Okay, so first we have the nuclear membrane. The nuclear membrane is a double membrane structure. It is composed of two membranes, two cellular membranes, and it separates the contents of the nucleus to the cytoplasm. The nuclear membrane is also known as the nuclear envelope. It is composed of two cellular membranes that separates the contents of the nucleus to the cytoplasm. Right, the nuclear membrane or envelope completely encloses the nucleus, like what I mentioned earlier, and separates the cell's genetic material, DNA, from the surrounding cytoplasm. Okay, serving as a barrier to prevent macromolecules from diffusing freely between the nucleoplasm, that's the protoplasm inside the nucleus, and the cytoplasm, that is the protoplasm outside of the nucleus. Then we have thread-like structures called chromosomes that contains our genetic material in the form of DNA. The chromosomes can easily be seen through an ordinary microscope, especially if the cell is undergoing cell division. These chromosomes are easily visible. These chromosomes, they contain our genetic blueprint or DNA. And also we have the nucleolus. The nucleolus is a densely stained structure, and the nucleolus synthesizes the ribosomal RNA and it assembles ribosomes.